Hello, and welcome to my CHEM 3511 Hot Topics video presentation, Weighing the Molecules of Life, Proteins. Proteomics is an up-and-coming field that studies the structures, functions, and interactions of proteins. Proteins, as you likely know, are biological macromolecules made up of different combinations of 20 amino acids. In comparison to genomics, proteomics is not as established due to the complexity and dynamic nature of protein expression. Much of this complexity comes from the fact that proteins are made up of 20 amino acids, but DNA is made up of only 4 base nucleotides. Another significant challenge in proteomics is post-translational modifications, or PTMs, which are not encoded directly in the genome, so they add a layer of complexity when you're studying proteins. The applications of proteomics are wide-ranging. It has its place in biomarker discovery, drug development, agriculture, food science, and many more. In the study of proteins, molecular weight is often a fundamental parameter for investigating, sequencing, and identifying proteins. Molecular weight analysis can help us understand the structure and complexity of proteins. It can help us discover biomarkers and also evaluate drug affinity and specificity. Biomarkers can be identified by comparing a normal and diseased protein sample. If there's a difference in molecular weight, that could correspond to a biomarker. Mass spectrometry analysis can also evaluate the affinity and specificity of a drug by monitoring for the mass of a drug-protein adduct and quantifying it by using methods like an internal standard. Over the last 20 years, advancement in mass spectrometry have opened up the broad scope of proteomics, and it has also become the method of choice in characterizing proteins. For an intro to mass spectrometry, MS is an analytical technique that detects and separates ions based on their mass-to-charge ratio. If we look at the block diagram of an MS, you can see the steps of an MS analysis. The sample is vaporized, ionized, it goes through an MZ analyzer, and then it goes to a detector, and then finally it goes to your computer where you'll see the data as a mass spectrum. Common ionization methods that you use for MS in proteomics is ESI and MALSI. And these are both soft ionization techniques that do not fragment the analyte as much as a hard ionization technique. ESI works by creating an aerosol of charged droplets in an electric field and desolvating the droplets to form gaseous analyte ions. MALSI ionizes proteins that are incorporated into a matrix material using a laser pulse. The presence of the matrix is important because it protects the analytes from fragmenting because it absorbs the excess laser energy. MS is a very useful technique, and these are some of its advantages. It has high sensitivity, resolution, and specificity, and it's a high throughput method, so it can provide data on thousands of proteins at once, and it can also run a lot of samples in a short amount of time. It's an especially strong technique in detecting peptide modifications that occur through processes like phosphorylation and glycosylation. Lastly, MS data is quite reliable and reproducible. On the other hand, the disadvantages of MS are that the sample preparation can be complex because often it requires steps like purification, separation, and enzymatic digestion. The instrumentation is, of course, expensive. MS also has low dynamic range, and it may not detect low abundance proteins if their signals are being obscured by higher abundance proteins. The data analysis requires specialized software, and the identification of proteins using MS depends on the database. You can't identify a protein without its characteristic fragmentation pattern and a database that has it. MS is now a crucial technique in proteomics, and the field currently relies on bottom-up MS for analysis of proteins. So the bottom-up approach requires this enzymatic digestion of the protein, and normally trypsin is used before you can do an MS analysis. The opposite approach is top-down, in which the protein is intact. It's not as common because it's less sensitive, and it also falls behind bottom-up MS in throughput and proteomic coverage. The top-down MS analysis also does not work for proteins that are larger than 50 to 70 kilodaltons because it's difficult to produce the fragmentation patterns. On this slide is an example of an MS-MS spectrum for a 690.39 Dalton peptide fragment, and this mass spectrum has used ESI. It's also a tandem MS, so the fragmentation has been done twice in this case. So this pattern that you see here should be characteristic, and we could match this pattern to something in a database and figure out what the protein is in this sample. For now, it seems like MS is going to continue to dominate in proteomics despite its challenges, but ongoing advancements will hopefully push the boundaries of what we can measure and what we can understand in proteomics. These are my references. Thank you very much.